Hey, that worked. Now, underneath all of this, does this work? Even better. Imagine that, things working. Missouri Association for Creation, yes, Marv is correct, the wonder of the cell. Zach, if I wander, is that going to mess up anything? Good, because I wander. That's me, and if you get a copy of this, I taught at Washington University for 25 years, and I have a Bachelor of Science in Systems, and that's about enough of that. Okay, so jumping right into it, you've all heard of Legos, I hope. Legos are good, and I have Legos. I'm not going to bring out my Legos. I am going to bring out some other things because Legos, in a sense, if you've ever gotten kids around Legos, what do? They build things. And in a sense, the Legos are the fundamental pieces from which we can build much more complex objects. These are examples of Legos for grown-ups, you could say, analogy, right? Kinds of Legos for grown-ups. And here's the kind of things grown-ups build, oil refineries and synthesis type processes and factories and so forth. I just love these pictures of oil refineries because they're impressive. No one would look at something like this and say, oh, what kind of accident occurred to create this functioning, amazing factory that does all the things it does. And so, yes, these oil refineries are impressive, and they are also simplistic compared to what we're going to see inside a cell, a single cell. So but it's easy to see here. So in a sense, what is God's Lego that he builds things with? Well, it's the cell, and it's on that poster that Marv's going to give away, so make sure you get one. There are two major classes of cells, what are called the prokaryotes and the eukaryotes. We're eukaryotes. Eukaryotes have a defined nucleus. And so that's what this is. This has a nucleus. Oh, unfortunately, my laser doesn't show up on the television, so you'll just have to hear me describe it. The purple thing, the purple round thing in the middle, you'll see it's a nucleus. I'll have several different pictures of this because there are many of them on the web. And I would call your attention to all these other little things that are labeled. Those are organelles, little other things for now in the cell. The nucleus has these little holes in it called nuclear pores. Those are important. We're going to talk about those a little bit, but we're also not going to talk about a lot of stuff. We'll see. So what's the theme for this? Well, here's some happy people. I praise you, Lord, because I am fearfully and wonderfully made. Your works are wonderful. I know that full well. And I have videos of figure skaters and gymnasts and remarkable things that humans can do. And it all boils down to what your cells can do. I've taken some of that out so we have more examples and other types of slides to talk about. So cells, like I mentioned, they're God's Legos. They're very tiny. And here's an example of a few different types of cells. We've got muscle cells off to the right. Lower left-hand corner, we have the kinds of cells that would be found in the blood. The blood, of course, has red blood cells, has white blood cells, has a number of other things that are labeled here. And in the upper left-hand corner, we have some glandular tissue, and it's something to think about. Cells, all these cells do different functions in your body. The cells that align your gut are secreting enzymes to assist in digestion. That's entirely different from the clear cells that make up the retina of your eye or other cells in other parts and functions of the body. And so there's a whole bunch of red blood cells. Here's something I like to, I'm going to just pop out here and see if I can find this in presentations. And let's go to the wonder of the cell. And there's going to be an HTML page here, cell size and scale. Let's double click that HTML. Let's open it with our favorite browser for the moment. And we'll pick Chrome. Like I said, for the moment. We may end up picking something else later. And so what's this slide about? Well, oh no, you can't see it. Let's see if we can change that. If I get out of this and slideshow, and I'll tab over. Can you see it now? Oh yes, you can see it. Okay, so I have two little baggies, and I may pass them around here in a little bit. They are, they have coffee beans in them. How many of you, coffee, a little coffee bean? I've got a little baggie with coffee beans. If you haven't seen it, I'll pass that around later when we get to a slow point in the talk. What I like about this slide is there's a coffee bean. Can you see it right in the middle? It's going to be hard to see from the cheap seats, but there it is. And what this says is how do you, how relative are the size? There's a grain of rice to the right of the coffee bean. Here's some measurements down here. A meter, how big a meter is, a centimeter, a millimeter, a thousandth of a meter, a micrometer, a millionth of a meter, a nanometer, which is a billionth of a meter, an angstrom, roughly 1 angstrom is roughly the width of a single hydrogen atom, and then even a picometer, which is smaller than that. Okay, so what this slide lets us do, watch what happens when I slide this. Oh, I'm drilling in. See how it's getting, I'm going in deeper. Okay, so let's go in. We're talking about how big is a cell? Well, now I'm down to amoebas. There's a human egg. The egg is a very large cell, and there'll be a sperm cell there. There's a skin cell. There's a photoreceptor. See the tiny sperm cell next to the egg cell? Let's keep drilling in. And there's chromosomes. We're going to talk about chromosomes as well. And there's Baker's yeast, a single cell. Now we're getting down into the organelles of a cell. Here's a mitochondria, a lysosome. Lysosome's like the garbage collector of the cell. E. coli, there's an E. coli bacterium per scale. Keep drilling in. We're now into the 45 nanometer realm. There's a hepatitis virus. There's a rhinovirus. But hey, there's more. Let's go deeper. And we keep going deeper and deeper. We get all the way down to the hemoglobin and RNA and antibodies and so forth. And you just drill all the way, and you finally get down to a carbon atom. So the point is, cells are small. They're made up of things that are even smaller. And if I could do this smoothly on my leg with my mouse, which is not, it's a little bit of a trick. Anyway, this website is pretty interesting. And again, I just stored off the HTML so I could show you that. That's how small cells are. Okay, let's press, let's set the mouse down so I can do this again. And of course, it took me to the beginning, but that's all right. We aren't that far along. And getting back to my other mouse, my signaling mouse, that's how small they are. Okay, so what do you need to see cells? Well, you need microscopes. These are light microscopes and different kinds, and they have makes nice microscopes now where you can get the attached video camera and so forth. This cell is a wonderful thing. So here we have an interesting slide. Animals and plants are both eukaryotic cells. They both have a nucleus, but here on the left side, they just blended one for the purpose of instruction. On the left side, you have a plant cell. On the right side, you have an animal cell. So a few things that are different on the plant cell, you'll see some different organelles, like for instance, well-known chloroplast. Of course, that's where photosynthesis occurs. Or the vacuole. Vacuoles are interesting. They maintain osmotic pressure in the cell. That's responsible for, if you see a plant with the leaves very healthy and kind of standing up, caking in the sun, they've got enough moisture. Well, that's because the vacuoles have enough moisture and water to maintain the osmotic pressure to keep all those cells. And it's part of a larger problem or challenge of maintaining rigidity and yet some flexibility. It's all at the same time. So humans don't have to do that. Animals don't have to do that. So we don't have vacuoles. We don't do photosynthesis. We have it's very similar but different set of organelles. And this particular cell also has off to the right there a red tail, known as the flagellum. That's not the bacterial flagellum, and the motor for this is entirely different. And the genes that code for it are entirely different than the bacterial flagellum. We won't go down that far. But so there's all these parts of this, of God's Lego. And let's drill in here. So what's so special about cells? Well, what can we talk about in an hour roughly, maybe 90 minutes max, but hopefully an hour and 15 or so. What can we talk about? Well, DNA, cell membrane, molecular machines, signaling. That's what I think I can get through tonight. That means I'm not going to get to metabolism, organelle, some of the details on organelles, except I'll mention in passing because I find this stuff just fascinating. We're going to see God's handiwork throughout this process. And repeatedly, we're going to see examples of what are called irreducible complexity, things that exist and function only if all of their constituent parts are working in unison toward the common function or purpose. We won't have time for self-replication, mitosis, amazingly complex process, very hard to create this one step by step, slowly over 1 million years. It either works or it doesn't, and this is how cells replicate. reproduction, which is a different but related form of cell division, meiosis, which also includes some gene shuffling so that all of your offspring don't just look exactly the same. All of your offspring look different, right? There are two different, at minimum, of two different stages during meiosis where the genes are shuffled, not completely, not enough to destroy that it's still a Homo sapien, but to change the eye color or to allow some other features to be brought forth, and such that every offspring, you could have 25 kids, and none of them will look exactly the same. You'll see similarities, of course. What else will we have time for? Stem cells and differentiation, various cell types. And I mentioned the cells in the retina versus the cells that line the gut lining or the stomach. Imagine the cells in your stomach that can withstand such a caustic environment that allows your food to be digested. Well, there's cells lining that. They do okay. In fact, they're probably secreting the enzymes that assist in that digestion. All kinds of different cells. That's just a remarkable thing. The cytoskeleton, that's what gives, like the vacuole for plants, all cells have, using microtubules and these other components, things that comprise the structure or give the cell its structure, its shape. And yet at the same time, cells have to remain remarkably flexible. I think I probably first encountered this in a talk that Scott did, but the blood cells, interesting. So remember we saw the red blood cells a little bit? They end up going through your veins, your arteries, into your capillaries. Capillaries are very, very, very small. How small are they? Well, at the smallest point, you could get 40 capillaries all wound together in the size of a human hair, the same diameter as a human hair. This assists with oxygen, with gas transfer and some other things, but it's actually smaller than the diameter of a red blood cell. Oh boy, how are we going to get a red blood cell to a capillary that's smaller? Well, this isn't the scientific term for it, but I did just hear a lecture on COVID that discussed this in detail. There's a squishiness element to red blood cells. They actually get squeezed down and they elongate and they disform a little bit so that they can fit through this smaller path, and then they reconstitute their original shape on the other side. It's pretty remarkable, and if that's a little bit off, you're not going to live, you're going to die. So, or in the case of COVID, actually it does have problems and it does lead to complications. That's one of the things that put people in hospital relative to the COVID infection. Photosynthesis, there's a topic we're not going to scratch anymore tonight, but imagine that would fill up a whole board and then another board with the chemistry behind photosynthesis. Homeostasis, that's maintaining equilibriums. There's pH equilibriums, there's temperature equilibriums, there's all these things, your blood pressure. Get it too high, that's a problem. Get it too low, that's a problem. All of these different aspects of your body and all the way down to the cellular level are maintained in equilibriums. That's under the topic of homeostasis, et cetera, et cetera, et cetera. So we won't get to all that in a short lecture about the cell, but that cell, it's been estimated by one scientist that a single cell, is more complex architecturally than New York City. Wow. The entire city. Yeah. Every light switch, every toilet that flushes, the subway system, the skyscraper, the whole bit, one cell, more complicated than all of New York City. We'll see, hopefully enough to get a glimpse of that. I'm going to start with DNA, but I'm not going to do a lot on DNA because that was part of that lecture I did a while back on the death of the neo-Darwinian synthesis and how mutation and natural selection are inadequate to lead to novel new structures and functions. Even the evolutionists are understanding that and proposing alternatives. But we do need to touch on DNA because it's a pretty critical component of the cell. And so it's at the center. We said that nucleus was important and those pores were important. We'll get back to that. So how does that happen? Well, here's a cell up in the sort of upper left middle, the kind of blue with the nucleus in the middle of it. And then it's just jumping and show you, hey, this chromosomes are organized and wrapped around these proteins called histones, which in turn, if we unwind all that, we get the double helix structure of DNA. DNA, of course, deoxyribonucleic acid, consists of these nucleotide bases, which are arranged in a sequence. And the analogy I like to use is this is like the roll on a piano player, of a player piano. Flip my words there, makes a difference, of a player piano. The DNA is sort of like that. Those are your instructions. If you get this piano roll, this might be Maple Leaf Rag, this might be a Beethoven sonata, but that's the roll. It's got all the notes on it, but it really doesn't do much for us unless we have the player piano. And what we're learning about cells is DNA is super important, but you can't go anywhere without the cell that it belongs to. I mean, there's some limited things you can do in the very single cellular organisms, and there's like Craig Venter, who extracted the DNA and they sewed in some different sequences and they got it back in, and they merged some yeast aspects with, put it back into the host bacteria. But really, it's the cell, which is analogous to this piano, the player piano, that is able to interpret and perform what's on the music roll. So that's how I like to think about DNA now. DNA is super important. Will you get a song if you don't have a roll? No. Will you get a song if you don't have a piano? No. This is really solving the chicken and the egg problem. When God created, and he said each after their own kind, it goes all the way down to the cell. I mean, the mapping of what DNA does is affected by what kind of animal or creature it's in. This is something that, well, I've not digressed too much there. What if we looked at the operation manual for a player piano, we see all of these diagrams of pieces and parts, and this is remarkably similar to what we'll see when we drill into components of the cell. There's all these diagrams and pieces and parts that carry on functions of the player piano. Again, if you remove 2 or 3 of them, it might play fine. It might have a clunker here and there. You remove the wrong one, if you don't have a source for energy, either plugging it in or pumping those pedals, it's not going to play the piano roll. So all the interdependencies of these components, we also find in the cell. These things don't exist on their own. They each have a super important role to play. And again, it's a little beyond what we can cover in one talk, but hopefully we'll get you a taste of this. So the DNA structure, wrapping up this point, the nucleotide bases that code for, what do they code for? Well, they code in these, what are called codons, sequences of 3 at a time. They code for amino acids. There's actually mapping tables. And I used to think, oh, this is the mapping table I got from a textbook. There's at least 30 different mapping tables. The mapping tables of some animals are different than the mapping tables of other animals. That's super important. If we all had a common ancestor, if we descended from a common ancestor and the DNA is just getting a little expanded each step, it wouldn't work if the mapping tables totally changed from this step to that step, such that what maps for guanine and tyrosine might be totally different in the next animal. So this is one of like at least 30 of these different tables. And here I show the 3-character sequence that maps to serine, if you can see the little red oval there. And of course, serine is one of the amino acids that we can chain together to make something like a lysozyme. Lysozymes in your tears. Alexander Fleming discovered this, I found out. He was leaning over, and this is a little disgusting, sorry. He's leaning over his petri dish in the lab, and a little bit of snot came out of his nose. Turns out it's in your lysozymes in there. It's in your tears, it's in a lot of your mucous membranes, and it killed some of the bacteria in the petri dish. And so he went on to discover lysozyme. It's full of these serines, as well as all the other roughly 20 amino acids that spell this out. So what do amino acids together make? Proteins. Proteins, as I mentioned before, made up of amino acids, and their shape determines their function. The example I like to use is car parts or any kind of nut and bolt situation. Here's 2 things. This is a nut and a bolt, and I have a hook. Its shape determines its function. It shouldn't be that mystical. The reason this nut works on this bolt is because of the thread pattern on the inside of the nut, the shape of the bolt, the distance between the threads, and the outside that allows me then to twist or not twist. If that was a rounded thing, it'd be a little harder to tighten down, wouldn't it? If you've ever rounded off a nut, you'll know that. And so the shape is determined from a process inside the cell, and its shape determines its function. And so therefore, we have a sort of a parallel between, oops, did I leave my cooker over there? I did. Glad it's not feeding back. So proteins, and I want you to remember this. There's a couple things to remember. Remember that little protein, because later on we're going to see a video and we're going to see proteins in action. And you're going to see this squiggly, colorful mess doing something. What's that? It's a protein. It's one of the whole set of proteins as part of a larger complex. There are complexes and chains, cascading chains of events that occur at the cellular level that involve 30 or 40 or 50 proteins in very specific arrangements and timing and distance, all kinds of precision at a scale that we can't match in the human world, but I'll get to a little bit more of that. Proteins are pretty, at least when you color them. They really, there's some artistic license in these colorings, but you can actually go to and some of these we've heard of, collagen. You've probably seen commercials. You should take collagen or something. Hemoglobin, the oxygen carrying protein molecule in blood, which is made up of a complex program. Anyway, what I wanted to point out to you is, well, where can you learn more about this stuff when we're done? We'll get the poster for sure. 2 books I would advocate. One is Design and Complexity of the Cell by Jeffrey Thompkins. I got 1 or 2 slides from him. They're pretty good book. And another one is Change Tan's book. I think I'm on my 3rd reading of the same book. It's technical and wonderful. 2 books that really flesh this out in a remarkable way. Okay, so here's just one aspect from the Thompkins book. DNA codes amino acids, right? A, C, T, and G, and there's these 3 character sequences mapped to this amino acid. The next 3 characters map to this amino acid. Next 3 characters map to this amino acid. Okay, fine. That's called within the coding region of DNA. So the DNA is unwound, and this is a word you've heard in the news lately. mRNA, messenger RNA, you've probably heard that recently. mRNA is extracted from, there's a synthase, there's a very complex molecule that assists in both unwinding the DNA and then allowing us to make a copy of it. And so, okay, fine. The mRNA will then go out to those nuclear pores, and where it'll travel to something like a ribosome, which sits on the rough endoplasmic reticulum and starts to make the protein, starts protein synthesis. This is all a big process. Well, okay, I kind of knew that, but then wait, there's more. Yeah, every level of this, there's more. So this is what Thompkins brings out. And if you look at the nature, at the structure of a protein, and you can't really see it, but there's a little tiny red sliver in the top bar there. That's just a section of a chromosome, and it's saying, hey, this is a gene. Now, what's been thought or believed for about 80, 90 years is that a gene codes for a protein, because there's a sequence of A, C, T, and G in the gene that spell out the sequence of amino acids. Gene codes for protein. We thought we knew that, but wait, there's more. Okay, what happens to that gene? Well, how's the gene made up? First of all, there's regions within the gene. There's the TATA box. Oh, what's the TATA box? That's TATAA. It's a sequence that appears at the beginning of the gene. Why? So DNA, we mark the beginning of a gene. We got to know where the gene starts. Is that magic? How did God figure that out? So there's a marker sequence, TATAA. Oh, wait, that's never happening anywhere else. So that means this is the beginning of a gene sequence. Okay, nice to know, but there's more. And then there's the exons and introns and components. So we have to, these are there for a reason. They get pulled out. And this is now what we're going to call pre-mRNA. So we're actually going to take mRNA. It's not ready yet. We have to process it. The cell has to process it. This is all still happening within the nucleus. So the pre-mRNA gets sections cut out. It may get other sections spliced in and manipulated. The pre-mRNA becomes something totally different than a simple mapping of what was in the DNA strand. That's what's fascinating, because lo and behold, you have about 20 or 30,000 genes. Well, I'll hold that thought. What I noticed when I was looking at this diagram is, hey, I've seen that before. These are TCP/IP packets that the data that you use on the internet travels down these packets. And these packets also have header sequences that mark the beginning of regions within the packet and tells it, hey, here's where the next message begins, here's where it ends. This is what we do in computers today. And yet we're finding that same kind of thing when we start to map out the DNA. When Bill Gates said the DNA is more complicated than any human program ever written, he was understating what we've got here. But here, how many genes, how many proteins? We've got maybe 20,000 genes, maybe 30,000. The number is even hard to come by. Guess how many proteins we have? Normally, I ask really simple questions. So if one gene maps to one protein, we have 20,000 genes. How many proteins we got? 100,000. Well, how is that? Well, it turns out that genes don't just get read in one direction. They get read in the opposite direction. Some genes are manipulated, they're sectioned and manipulated, the introns and exons and such, that the shuffling that you saw in the mRNA processing, before it becomes the mRNA that leaves the cell through the nuclear pore, goes on to the ribosome to become a protein. Well, now, because of the ability to manipulate along the way, this puts it pretty well. And this is from an evolutionary source. The human body contains roughly 20,000 genes that are capable of producing proteins. Each gene can produce multiple forms of a protein, and in turn can be decorated with several post-translational modifications. The number of potential molecules you can make from one gene is huge, says Bernard Kuster, who studies proteomics at the University of Munich in Germany. It's very hard to estimate, but I wouldn't be surprised to have one cell type 100,000 or more in a single cell type. So that may be in a muscle cell or in a single skin cell, to have 100,000 or more proteins found there. So how does that happen? God's a master programmer. That's how that happens. And the thing about programming at this level of complexity, any random change to that code destroys it. When we look at the regulatory, they can manipulate some aspects of DNA and say, hey, look, the mutation. They're not going to grow wings. They're not going to do anything useful or beneficial. They may make it, help it metabolize something it couldn't. Bacteria may be able to metabolize something it couldn't metabolize before. But you're not going to enhance the quality of the code through random changes. Every programmer in the world knows this, and it becomes more true the more we learn about DNA. Okay, on to the next topic, cell membranes, which if there was, you could get multiple PhDs on different aspects of cell membranes. This is a fascinating thing. Now, the membrane is this basically the liner around the outside of the cell. And if you look at the basic, hey, how did this evolve textbooks, they'll say, oh, you know, this is what's called the phospholipid bilayer. You have these hydrophilic heads and hydrophobic tails, and they arrange opposite each other so that what's on the outside of the cell and on the inside of the cell, outsides at the top, insides at the bottom, that has an affinity for water. That's what hydrophilic means. Philic is in love. But hydrophobic means I hate the water. I'm a water big and I don't like water. The tails are hydrophobic. And this arrangement, what this allows us is it creates a membrane. Well, lo and behold, they have found these things can be formed naturally in a petri dish or in a test tube. And they said, aha, first step toward creating cell membranes. This is one of the fundamental things you need to do to create life. Well, do they have something there? And the answer is no, of course. It turns out no cell on the planet could survive a moment if this was its membrane. The membrane won't work. What makes cell membranes work is stuff like this. They are semi-permeable membranes. They have to take in nutrients, take in gases. They have to pass out waste products and other byproducts of metabolism. Without that semi-permeability, cell membranes would just kill the cell. It'd be like put you in a bag of plastic, see how long you last. It's just not going to work. So it's way beyond what you need to just form these nice lipid barriers with the heads and the tails. It gets deeper and deeper. And I thought, well, how can I scratch this topic? And so I'm going to go to Change Tan's book for this. This is a quote from her book that I found pretty interesting. I'll break it down. One of the essential requirements of a membrane is to maintain a higher concentration of protons on one side relative to the other. That is a proton gradient. We're going to find out that there are machines in the cell, that's in the next section, that are not electron powered. We power stuff with electricity. We can manipulate and work with electricity. We store it in batteries, all kinds of neat stuff. A lot of these cellular machines are powered by proton power. And that's a level of fascination that is another chapter in her book. It gets into quantum mechanics and into some of the most remarkable aspects of physics that we still don't fully understand, but this is how we see it working. Anyway, so this proton gradient is, the idea here is, it's like a dam. Imagine what the dam does to a river. The river comes up and gets behind the dam. And now you have a water pressure gradient. You got a lot of water pressure sitting at the dam. And on the other side, not so much. And then what do? We open a little hole in the dam and let the water fly through and power a turbine for us. And so it's useful to have this gradient, this difference in pressure. In the case of the cell, we're going to find the pressure is protons. We're going to find we have a gradient with a high concentration of protons on one side and low concentration of protons on the other. Okay, hold that thought. If the membranes in your body suddenly lost their proton gradients, you would not be able to finish reading this paragraph and your life would be unrecoverable within minutes. That's how cyanide poisoning works. It kills the proton gradient capacity of your cell membranes and you die. Cell membranes have to maintain the proton gradient. They have to be semi-permeable. They have to let things go through. But you know, protons are really, really tiny and it can't let them go through. It has to maintain a gradient. More protons on one side of the barrier than on the other. Hold that thought. A nonspecific membrane pore, just think of a hole. If I just pop a hole in a cell, I need a pore. Okay, pop a hole into a cell. A nonspecific membrane pore that is always open would destroy the proton gradient. Hopefully that's understandable now. Therefore, pores must be highly specific, and most pores open only when triggered to open. Well, that's part, just like our gate on the dam. We decide when we let the water out. That's good. Aquaporins, guess what they allow to go through? Any guesses? Water. Yeah, cells need water. We need water. It works. Cells take in water. Cells give out water. I was listening to a neat lecture. I would like to do a lecture just on water. As a base molecule, water has some temperature retention properties, some heat retention properties that make it amazing. It holds heat long enough that when a water molecule is passed out of your cell, it's still holding heat. It's a way to transfer heat from the cell, because when you need to cool off, if you're overheated, it's very important that you don't get too hot, don't get too cold. And water plays an important role just down to the actual physics of the water molecule. It's fascinating, but anyway, aquaporins are pores in the membrane that are going to let water go through, okay? A specialized membrane pore that allows water transfer across the membrane. Aquaporins allow water to pass through the membrane. Here, remember this. A water molecule is bigger than a proton. Keep that in mind. Allow water to pass through the membrane while blocking the passage of protons. This occurs by precise manipulation of each water molecule, such that each molecule is rotated perpendicular to the orientation that would be required for the conduction of the protons. And so, in other words, at the molecular level, we have the cell membrane manipulating. There's these chaperone proteins that allow these things to be passed through the membrane and yet maintaining the ever essential proton gradient. More protons on one side than the other. Anyway, cell membranes are fascinating and involve signaling, which I'll get to in a second. But let's go on to molecular machines. And we have our first video. See if I can make this work. I started with a simple example. I think this one is the Kinesin Motor. And I can get there in a second. Let me give a context for here. Okay, so you remember you got the nucleus and you have all those organelles, and you're going to be creating proteins. The proteins are pretty much started at the ribosomes, carry out the RNA transfer into amino acids and the beginnings of proteins. There's more stages that have to happen. Proteins have to be folded into a shape that makes them serve their purpose. Remember, their shape is ever important, or a protein to work right, it's got to have the right shape. So there's other organelles that help with that. But now let's say, hey, I've got the perfect protein to chaperone an aquaporin across the nuclear membrane. I need to get that protein from where I just made it here over to that cell membrane over here. How are you going to do that? Well, there's a transport mechanism. The transport mechanism involves these little highways and little trucks on these little highways that move proteins around as needed. The highways are constructed and deconstructed as needed, although there are targets on the inside of the cell membrane. We don't fully understand all the ways that the highway knows. How do you know where to build the highway? The cell knows. It builds the highway to the right spot to deliver. So there's signaling that's going on to say, hey, I need the proteins necessary to do water transport across the cell membrane. And so there's these proteins being manufactured, they're being transported around the cell, they're signaling saying, hey, what I need has to be responded to. So this is the Kinesin Motor. And what's fascinating, well, I'll show the video and then I may even stop for questions if I can get my, let's try this mouse. I have a mouse cripple. There we go. Is that gonna work? Yeah. Oh no, give me more, give me more. if it won't give me more, I won't be thwarted that easily. Let's do, this is my, let's pop back over to that folder where I have all this stuff, and let's look for
inside a living cell is a food transportation system. Proteins have to be delivered to the correct part of the cell to perform their intended functions. This animation, based on a lot of clever research over a number of years, shows how it happens. Highways made of microtubules are assembled by interlocking proteins, each manufactured in accordance with the coded instructions on the cell's DNA. Marching along a microtubule is the Kinesin Motor, the hero of our story, carrying a huge sack of proteins to be delivered to a predetermined place in the cell. Here, the proteins will be released to fulfill their functions. A kinesin linear motor uses 1 ATP to provide the energy for each step and takes 125,000 steps to cover 1 millimeter. This amazing machine shows all the hallmarks of design.
Okay, that was the simpler one. Let me do the more complex one. This one is from the ID, folks, but it's very similar.
Cells are full of specialized components that perform functions vital to their existence. But how do these components get to the right locations inside the cell to perform their functions? For larger components, a transportation system is needed. Meet the kinesin. Masterpieces of microengineering, kinesins are miniature motorized machines that carry cargo from one part of the cell to another, walking along self-assembling highways called microtubules. Known as the workhorses of the cell, kinesins have 2 feet, or globular heads, that literally walk 1 foot over another along the microtubule, pulling their cargo to its destination. Each foot possesses 2 special locations called binding sites that interact with other molecules. One site attaches to the microtubule and the other binds with ATP, the energy molecule of the cell. When one foot binds with ATP and uses its energy, the foot flips over, resulting in the walking motion. Each foot has a short neck, which is connected to a strand of a long coiled stalk. At the end of the stalk is a fan-shaped tail, which holds tightly to the cargo being transported. Kinesins can carry cargo that are many times their own size. Sometimes a kinesin is in danger of getting stuck on the microtubule highway because of blockages caused by other cellular components. To get around such obstacles, multiple motor proteins may be used to carry a single piece of cargo, together providing enough force to break free. Kinesins typically walk away from the center of the cell and toward the cell's periphery. The kinesin's 2 feet work together efficiently, with 1 foot holding fast to the microtubule, while the other releases itself and takes a step forward. This coordinated stepwise movement allows Kinesin motors to walk as many as 100 steps per second, moving about 8 nanometers with each step. When not carrying cargo, kinesins can shift to energy-saving mode to conserve fuel until their next job. The kinesin plays a vital role in many cellular processes, not just transporting materials, but also aiding cell replication. The walking kinesin molecular machine, another example of intelligent design.
Okay. Let's see if I totally lost my, oh, I think it'll work. So here's a couple of things that you don't necessarily see the first time through. First of all, the highways get built, constructed, deconstructed. Why? If all those highways stayed in place all the time, the cell would get too rigid. And that rigidity would be a problem. As we mentioned, think of what red blood cells have to do, although that's a entirely different situation. But most cells have to retain some flexibility. It's nice that you can, press on your skin and not have it all rigid. But think about those tubules. As they're constructed, they're round. And at first it didn't occur to me, why a highway? Why would you have a round highway? It's like a straw. Why not just a flat thing? Well, for maximum ability to attach kinesins to the problem load, you would want to, the most efficient structure would be round, because remember, those 2 feet would fit around this thing. The next one would fit next to that. The next one, if it needs a third one, would fit next to that. The next one, if it needs another one, would fit next to that. And so by constructing a tubular highway, you maximize the surface area that the kinesins can attach to drag that load along. Nice little feature, dumb luck evolution to come up with that. I've got a third presentation that's from Harvard, and it's just more technical and more about the chemistry and the ATP. ATP, which we won't talk much about it, adenine triphosphate. We're going to find that ATP is the energy molecule of the cell. And as it's used up by components within the cell, it becomes ADP, adenine diphosphate. Well, it goes from 3 phosphate ions down to 2. That release of that third phosphate ion is sort of like the little step or little punch, the little strength. I left it out because I thought, nah, I can't get into all that. And I can't speak about it without my notes because it is complicated. So the point being, ATP, you'll find is the energy molecule, and then you'll see ADP. Well, that's after it's spent, the phosphate ion is spent. Now it's ADP. That's going to become important for another video here in a second. But back in the Harvard video, the one that they did, the best part of that one that I thought about using it. So there's a little parking lot. Where do all these Kinesin motors sit when they're not being used? They all go park in this little parking lot. That's the rest mode. They kind of just wait until they're called into action, and then they come out of their little parking lot, hop on the tubule that's sometimes being built right in front of it as it's walking along with the load that it was told to carry out to some point in the cell. But there's no design here. This is remarkable evidence of design, and I just find it terribly exciting. Let's look at another molecular.
I'm sorry, Steve.
Yes, sir.
How's the tube know where it's going?
Okay, it's a good point. Yeah, but there are what are called membrane targets that we don't fully understand. If you recall, I briefly mentioned on one slide, I had one bullet point on membrane targets. It's in the world of epigenetic information. There's a lot of stuff, information and knowledge in the cell that is apart from DNA. Things that DNA has no control over, like the whole splitting of the chromosome is governed by some intelligence that's not DNA managed, because it's the DNA itself that's being split. Same with these membrane targets, and there's 2 or 3 related things, all part of the tubular highways that get constructed. And yeah, it's complicated, and we don't fully understand it, to be honest. This next slide, which I am going to show you a slide from a secular source. I appreciated your honesty. We don't know how this works. You're going to see that here in a moment. Okay, well, so let's do the simple. This is ATP synthase. So ATP, as I mentioned, is the energy molecule of the cell. ATP is manufactured by one of those special organelles. And anybody know which organelle manufactures our ATP for the cell? Mitochondria, right? That's one that you may have heard of, mitochondria. Mitochondria is remarkable for a number of reasons. It has its own DNA. That's how they got that whole mitochondrial Eve thing. And because it's totally different than the DNA that is inside the nucleus of the cell. Why does it have its own DNA? Well, because the production rates are so high on the manufacturing of ATP that there's no time to send off and, get me another package of proteins using the Kinesin motor so I can gear up and produce some more ATP. The whole mechanism for producing ATP is very tightly managed and part of the mitochondrial world. That's what this is going to get into. This is going to get into a molecular machine called ATP synthase. We've only discovered this one in the last, 10 or 20 years or so. And we're still trying to understand it. The guy's going to say, we don't understand this part yet. But what's neat about it, you're going to see it's embedded in a membrane, one of those phospholipid membranes, and it's the mitochondrial membrane. What you're going to see, maybe not in the first one, but definitely in the second one, is the proton gradient. You're going to see all these protons floating around on the inside of the cell and not so many on the outside. So it's a really good visual for that as well. Let's jump to the high-level ATP synthase.
This animated sequence shows the ATP synthase enzyme in operation. The animation is based on an incredible series of scientific discoveries. Only the colors show artistic license. ATP, or adenosine triphosphate, is the energy currency of the cell. ATP is produced by a tiny molecular rotary motor, rotating at up to 7,000 RPM. These are so small that 100,000 would fit side by side in a millimeter. A current of protons drives the motor, unlike man-made electric motors which use electrons. This portion of the enzyme is where adenosine diphosphate is combined with a phosphate ion in the presence of a catalyst to produce ATP, which is then released, making way for the next cycle. A top view of the enzyme shows the sequential operation. Almost every biochemical process in your body requires ATP. Such a nanomachine exhibits all the characteristics of super-intelligent design. ATP is vital for life, and many of these motors were needed before the first living cell could exist. An evolutionary impossibility.
So before I go back to my slide deck, I want to show you the next it's only 5 minutes, the next ATP synthase video, which is from a secular source. It's more complicated and it's going to go over our heads a little bit, but I think that's okay. You'll see the remarkable handiwork of our creator, which is why I'm doing it.
We haven't seen that already.
Yeah, in case you missed it.
You said that's the energy currency
Of the cell. Yeah, all those organelles, all your cellular functions that do anything really require ATP as the energy molecule. It's like a little battery that floats around. And again, recall that ATP is triphosphate, adenosine triphosphate. ADP is diphosphate too. So it goes from 3 phosphate ions down to 2, and that's the release of that phosphate ion is what uses the energy. And so now we need a machine to stick a phosphate ion back onto ADP to recharge it. It's like recharging the battery. So we need to create ATP. And this is happening, like he said, 7,800 times a second. I forget if it's in this video, but they actually know how many proteins it takes to cover each step of that little rotary motor to do each piece of the process. But this you'll give a glimpse of it here.
We are looking at the turning gamma subunit and the epsilon subunits, and there's the C ring in the membrane. The F1 complex is outside of the membrane, while the F0 complex is embedded in the membrane. There's the stalk, including the delta and the B subunit. The alpha subunits are shown in light pink. The beta subunits are magenta. In the middle, the central stalk is rotating. The B subunit stabilizes the hexamer, which means that the hexamer can't rotate. We will now look at a rendering of known structural states and the conformational changes that are happening. The beta subunits are seen as a ribbon diagram. The ADP and inorganic phosphate diffuse into the active site, and the ATP has just been released. And simultaneously, the conformational change occurs in the beta subunit. If we zoom into the active site, we can see the molecular structure of the substrates and the chemical reaction. Here is ADP with its 2 phosphates. They are all held in place by a network of hydrogen bonds mediated by the amino acids in the folded structure of the beta subunit. Hydrogen bonds stabilize the base and the sugar, but really primarily the base. There's also a magnesium ion that plays a critical role in not just stabilizing, but also neutralizing the charge of the phosphates. Notice the conformational changes that are happening when the inorganic phosphate comes in. There is the promotion of a planar transition state. synthesis of a new phosphodiester bond is catalyzed, and then the subunit switches to the open conformation. Upon this reconfiguration in the beta subunit, ATP is ejected from the active site. The enzyme then resets and restarts the synthesis of another ATP molecule. This is a summary of the events at the active site. It is also intriguing just how the C-ring rotates. We can start with the analogy of a water wheel. We can easily grasp the idea of how a water wheel rotates. It's driven by the flow of water, allowing the wooden fins to go in a particular direction. Thus, the rotation happens in a specific direction. If the water was to flow in the opposite direction, you would see rotation of the water wheel in the opposite direction. Here we see the C-ring and the A subunit. We don't know the full structure of the A subunit, but we know that there's an access channel for protons on the A subunit right in the intermembrane space. And there's a separate egress channel for protons on the matrix side. In this animation, you see protons flowing out from an interface between the C-ring and the A subunit. But what drives the specific direction of this rotation? For example, if we were looking down, it is always a counterclockwise rotation. Why and what drives that? On each of the C subunits within the C-ring, there's a proton binding site. Notice the high concentration of protons on the intermembrane side and their flow to the lower concentration on the matrix side. Protons load from the highly concentrated intermembrane side onto the C subunits, and they are released on the matrix side where the concentration of the protons is low. But why can't protons go around the other way as well? In principle, just as in the water wheel analogy, the C-ring should be able to rotate in either direction, shouldn't it? There are two interconnected reasons for this unidirectional movement. The amino acid side chains in C subunits would repel the bound proton, so that when a proton loads from the intermembrane side, the C-ring cannot rotate back in the opposite direction because it is energetically unfavorable. Rotating counterclockwise, on the other hand, is energetically favorable because there are no repulsive interactions. Remember, there's a concentration gradient that favors binding of protons on the intermembrane side and the release of protons on the matrix side. So in summary, the proton gradient determines the direction of the rotation, favoring movement in the counterclockwise direction of the C-ring. This avoids unfavorable interactions with the amino acid side chains of the C-ring.
There you go. So you got it was a nice visual of the proton gradient there. Do you see all those protons down at the bottom and not so many up at the top? That's what's being channeled to spin the C unit, the C subunit, and cause the whole process. I also liked where they mentioned the hydrogen bonding sites for ADP. If you're gonna, if you were in your shop and you want to glue two pieces of wood together, what do? You use a clamp, right? You have to hold something stable to work on. You put it in a vise. Those are like the little vices that hold the ADP just so that we can stick this phosphate ion on there. And we got to have that little magnesium piece there just to change so the electrical charges don't get interfere here. I mean, this is remarkable engineering at a scale that's so tiny. Like you said, 100,000 fit side by side in a millimeter. Yes, sir.
In order to compose these illustrations, are they observing this with mega microscopes, or are they observing it with chemical reactions, or how are they doing this?
Well, electron microscopes that's the neat thing about Behe's book. You can actually see electron microscope images of some of these motors, these molecular machines in action. They look, that last video was nice because they do kind of look like blobs of stuff doing blobby things. But it's only when you get into the details that you realize that's not just a blobby thing there. This isn't splashing around in a mud puddle. This is highly, there's a word, not managed, very specific and highly coordinated. And there's a third word that still escapes me. Yeah.
How many designs did the creator have to come up with before he selected the best one?
Well, you can ask him someday.
I think he did it just like that.
I just finished reading Job again, and I think that may be the answer. Let me know when you can tell him. because, yeah, where were you when I did these things? Yeah,
I think you got it right the first time there.
So back to the slides, and I did have a few more. So let's see here. Can I do the function shift F5? Would that work?
You know, it's hard to think that any
Nope.
real intelligent scientist would think this all happened by a big explosion.
It's a spiritual problem. It's not a function of intelligence, I believe. And yes, because you're right. It's hard to believe you can see these things and not see God's handiwork and be amazed and just want to worship your creator. I mean, that's one reason I love this stuff. It's just so inspiring. You think God's not in control? You know, the Bible has to put things in first century terms. So yeah, the hairs on your head are numbered because they didn't have microscopes yet. They didn't have electron microscopes yet, and they didn't know about ATP synthase yet. But yeah, God is in control of some remarkable stuff. These things, the rates of these things are occurring in millions of times a second. All of these metabolic processes occurring in your cells, they keep you alive. And if they fail, you're not gonna make it. And that's something to thank God for too. And we'll never even understand it till we get to the other side. Anyway, this was a picture of that ATP synthase. Here, this one shows the proteins kind of the structure on the right hand side. This is sort of the functional diagram on the left. This is from Change Tan's book. And I forget if I have a quote from her on this. Let's see. There's those two. Hey, it was working that time. Oh well. No, I guess I took out that quote because there's a remarkable. I will loosely paraphrase it. Get the book. I recommend it. But it gets into what's called electron tunneling. How do we get to those protons down in that gradient? And there's this 3-step process that breaks down carbohydrate energy to create the protons. And it does so it's like a cascade. She likens it to a slinky. Imagine a slinky going down steps. And it looks neat in the commercial. And so you go out and buy your kid a slinky and you put it on the top step. It goes about 3 and then it stops. Because, you know, everything has to be just right for the slinky to really make it all the way down the steps, all the way to the bottom. Well, this cascading process, and I forget, it's all described in chemical terms, very complicated. The distance between the steps is measured in angstroms, about the width of a hydrogen atom. If you get one step more than an angstrom more out of step in distance from the preceding or the next step, it stops working, just like that slinky on the stairs. The level of precision here is unimaginable. And so through this 10-step, highly precise process, we end up converting the energy that is in the foods we eat, in carbohydrates and things like that, into the protons that are needed on the heavy proton side of that proton gradient. She goes through the chemistry of that, and it's remarkable. I just didn't think I could cover it in a way without just reading paragraphs of complicated stuff. So I decided I would move on. Signaling. That's the last thing we'll talk about for tonight. Hopefully we're doing okay on time. Yeah, I think so. I never know. This is the first time I've done this version of this talk, so I never know how long is that going to take. Hopefully if it's 10 o'clock, they'll let me know. But it's not. We're doing pretty good. So this is one of the most complicated chapters of Jeffrey Thompkins' book, Cellular Signaling. And it's remarkable. It involves a lot of things we are still figuring out, like the sugar code and information sources and epigenetics that we don't fully understand. But just think about as I already mentioned, the Kinesin Motor is taking a protein from where it was manufactured to where it is needed. How did that information how was that signaled? Hey, I need this. So there's intracellular signaling. Hey, I need this protein. Hey, I need that. Hey, the lysozyme, the little organelle, the little trash collector organelle, he has to know which things, and they're marked so that he knows which things he can get rid of and break down and destroy. If he starts destroying the wrong thing, it's one of the many diseases that can kill us. So there's signaling that goes on inside the cell, but that's fine, but that's totally insufficient. Imagine your muscle cells. Okay, a muscle cell has all the signaling it needs to maintain itself, maintain its all the necessary homeostasis functions, to maintain the appropriate temperature and pH levels and all that. But how good is a single muscle cell? Really, your muscles are good because they work together. I mean, how do you, this is what we want to do. And that means I need all the muscle cells that are attached to those tendons to be able to do that. And so that's signaling. There's signaling going on to say, hey, muscle cells, hey, all act together. Well, then you have to get, that takes you up to the next level, which is the macro level signaling. So I decided I would do this. And I didn't get to warm James beforehand, but here he goes. Good catch.
It was almost intercepted.
It was almost intercepted. You know, Marv gives a great example of Isaac Bruce going out for the pass, and Kurt Warner's nailing this great pass, and it's all happening in like 2 seconds time. Just imagine the signaling that just had to occur. His eyes had to see that. That was an electrical impulse. His brain had to convert that. He had to tell his muscles, hey, I need to put a hand up here. The muscles had to know exactly when to close to catch the ball. It didn't just hit his hand. You know, so how remarkable is the macro level signaling that is controlling the interactions of all these cells to give us coordinated movements?
He needs to know if he's 50 yards down the field, what angle, the speed of his arm, the angle to let go of the ball. He's processing all this. He's watching the speed and trying to figure out where he's going to be. I mean, think about it.
And your cells are doing what they have to do.
Automatic.
Once your brain knows what it wants to do, your cells are kicking in. Yeah, so you have your autonomic nervous system, which is also telling cells, you know, you're digesting food right now. You're not thinking about that. You're breathing right now. So I was doing my morning walk up the big hill this morning, and I start breathing harder. First of all, I noticed that my muscles start to get a little sore because it's a really steep hill. And then I find my breathing harder. And then I find my heart's pumping faster. Well, imagine all the intercellular signaling that is going on. I got muscle cells saying, hey, I need a little more air here, a little more oxygen, maybe some more nutrients. Other cells, so your bloodstream is carrying signals. You've got now your thymus and some other glands kick in and start changing hormone levels, and things start happening there so that other functions within your body start reacting to those signals that are going on. Certainly your respiration increases, your heart rate increases, because if you're going to breathe in more air, well, you got to carry more air in your bloodstream. So they're coordinated. They have to be. And so you have a central nervous system that is handling all of those internet signals. And I'm not even thinking about that. I'm listening to my podcast. I just notice I'm breathing harder and my body's taking care of getting me up the hill. So the macro level signaling this is all astounding stuff. And again, it was probably the most complicated at the cellular level. Just the cellular level signal is one of the most complicated chapters of Thompkins' book. So yes, so what verse would I conclude with? Great is the Lord and most worthy of praise. His greatness no one can fathom. One generation commends your works to another. They tell of your mighty acts. They speak of the glorious splendor of your majesty, and I will meditate on your wonderful works. They tell of the power of your awesome works, and I will proclaim your great deeds. So I use that as my closing verse, and that's all I have to say about cells.